T.S of Mature anther. Transverse section of mature anther reveals the presence of number of anther cavity surrounded by an anther wall. It is a bilobed in structure and two theca is present, that is called dithecus. So it is consists of the three main parts. One is anther wall, anther cavity and a connective. So first one, anther wall. The mature anther wall consists of the following layers: epidermis, endothelium, middle layers and tapetum. So first one, epidermis. It is the outermost layer, single layer. It is the functions of outer layer is protections, and these cells are undergoes repeated in anticlinal divisions to produce the inner tissue. Next, endothelium. Next layer, endothelium. It is generally a single layer or radially elongated cells found below the epidermis. It is present below the epidermis, and this inner tangential wall develops the bands-like structure. It is made up of cellulose, sometimes lignified. These cells are hygroscopic in nature. And the cells along with the junction of two sporangia of an anther lobe lack of these materials, lack of these thickenings. This place is called, or this region is called stomium. This region helps to releasing the pollen grains at the maturity times. Next, middle layer. Two or three layers of the cells next to the endothelium constitute the middle layers. It is generally short-lived. At that maturity time of anther, it is disintegrate. Next, tapetum. It is the innermost layer of the anther wall, and it is derived from partially from peripheral wall. Periphery means outer side, peripheral wall, and then partially from connective tissue of the anther lining the anther locules. The tapetum is dual in functions. One is it nourishes the developing sporogenous tissue, microspore mother cells and microspore. Next is the sporopollenin, pollen kit. These are the minute spherical body-like structures. It is made up of that anther wall. Tapetum also controls the fertility or sterility of the microspores or pollen grains. Next, there are two types of tapetum. One is secretory tapetum and invasive tapetum. What is secretory tapetum? This tapetum retains the original position and cellular integrity and nourishes the developing microspore. Next, invasive tapetum. These type of cells lose their inner tangential wall and radial walls and the protoplast of the tapetal cells and all together to form a periplasmodium. Next, functions of tapetum. It is a very important 3 more questions. Functions of tapetum, it supplies the nutrition to the developing microspore, and it contributes the sporopollenin through the hibiscus bodies. That is a very important 1 more question. Through this hibiscus bodies, the sporopollenin is contributed. It plays an important role in wall formation. Next, the pollen kit material is contributed by tapetal cells and is later transferred to the pollen surface. And XII proteins responsible for rejection reaction of the stigma are present in the cavity of XII. These proteins are derived from tapetal cells. Next, anther cavity. And the anther cavity is filled with microspore in young stage. On this maturity times, its name is pollen grains. The meiotic division of microspore mother cells gives rise to microspore, whereas this haploid in nature. Usually the meiotic cell divisions, the end of the result is haploid cells. So haploid microspore will produced from the microspore mother cells. And then connective, it is a connective tissue. It is present in that place of this connective tissue. It is the column of that sterile tissue. Sterile means malatuthanmai. Sterile tissue surrounded by the anther lobe, and it possesses the vascular tissue. Also, it contributes this inner tapetum. Next, microspore and pollen grains. Microspore is a young stage, pollen grains at a maturity stage. You know, pollen microspore. Microspore is consist of the two outer layer, XII and intine. Then inside is cytoplasm. Inside the cytoplasm is consist of nucleus. So microspore are the immediate product of that meiosis of microspore mother cell, whereas the pollen grains derived from microspore. The microspore have protoplasm surrounded by a wall which is yet to be fully developed. Next, this pollen grains is consist of the two layers, outer walls is called XII and intine. The intine is made up of pectin, hemicellulose, cellulose and callose together. It is made up of proteins, made with proteins. Then exine the outer layer. The outer layer is made up of cellulose, sporopollenin and pollen kit. So this exine is not uniform and is thin at a certain areas. Next, pollen grains. The shape of pollen grains is varies from species to species. It may be globose, ovate, elliptical, fusiform and crescent. The size of pollen varies from 10 micrometer to 200 micrometer. So this wall material is made up of mostly pollen kit. Pollen kit is contributed by the tapetum. So this outer layer is made up of the pollen kit. It is colored in nature, maybe yellow and orange color. It is chiefly made up of this carotenoids or flavonoids. It is an only layer, oily layer. Thick viscous coating is present over the pollen surface. It attracts the insect and protects the damage from UV radiation. Next, development of male gametophyte. These are the stages of male gametophyte. The microspore is this first cell of the male gametophyte and it is a haploid in nature. The development of male gametophyte takes place the microsporangium. So inside the microsporangium, microspore mother cell is present. The nucleus is dividing into two cells. One is vegetative cells and generative cells. A wall is laid around the generative nucleus, resulting the formation of two unequal cells. A large irregular nucleus bearing with abundant food reserve is called vegetative cell and a small generative cell is formed. This is a two-celled stages. Next, this intine is grows as a pollen tube through the germ pore. In case the pollen is liberated two-celled stage, the generative cell divides into two male gametes. This is the stages of male gametophyte. Next to the development of male gametophyte, female reproductive part gynoecium. You know very well the gynoecium. The gynoecium represents the female reproductive part of flower. The word gynoecium represents one or more pistil of a flower. The word pistil refers to ovary, style and stigma. What is pistil? Where it is derived? A pistil is derived from carpel. The stigma serve as a landing platform for pollen grains. This place is landing platform for pollen grains. The style is an elongated, slender part beneath the stigma. The basal swollen part of the pistil is ovary. The ovules are present inside the ovary cavity. And the ovules or megasporangia arise from the placenta. Placenta means the basal part of the flower. The number of ovules in an ovary may be one or many. So the number of ovules present in the ovary, it is classified into one or many. For one means one example paddy, wheat and mango. Many papaya, watermelon and orchids. These are the example of that ovules present in an ovary. Next is structure of ovule. This is the structure of ovule, megasporangium. This megasporangium following these parts: salicylate, integument, nucellus, embryo sac present inside the nucellus, hilum, micropyle, raphe, funicle and vascular supply. Now you are going to see that explanation of that structure of ovule. So ovule is also called a megasporangium and it is protected by a 1 or 2 coverings called integument. This is the integument. A mature ovule consists of a stalk of a body. This is stalk. The stalk also known as funicle. It is present in the base of the ovule. It is attached with the ovule to the placenta. The point of attachment of the funicle to the body of the ovule is known as hilum. So it represents the junction between the ovule and funicle. Next, the ridge part is present in that ovule is called raphe. The body of the ovule is made up of central mass of parenchymatous tissue is called nucellus. The main function of the parenchymatous cell is storing the food materials. The nucellus is enveloped by the 1 or 2 protective covering is called integument. The integument encloses the nucellus completely excepted that micropylar region. So the ovule with a 1 or 2 integuments said to be unitegmic or bitegmic ovules respectively. The basal of the body of the ovule where the nucellus integument on the funicle meet or merge is called salasa. This is a salasa. There is a large ovule sac-like structure in the nucellus towards the micropylar end is called embryo sac or female gametophyte. This is a embryo sac also known as female gametophyte. It develops from the funicle. It develops from the functional megaspore formed within the nucellus. Next, what is endothelium or integumentary tapetum? In some species, the inner layer of the integument may become specialized to perform the nutritive function for the embryo sac is called endothelium or integumentary tapetum. So, these integumentary is given a food for that embryo sac. This is called endothelium or integumentary tapetum. Next, tuninucellate. If the sporogenous cell is hypodermal with a single layer of nucellar tissue around it, the ovule have very small nucellus. So crassinucellate. What is crassinucellate? Ovules with a subhypodermal sporogenous cell is called crassinucellate. Here the ovule have fairly large nucellus. Next, hypostase. What is hypostase? Group of cells found at the base of the ovule between the salasa and embryo sac. You know very well where it is present the salasa, where it is present the embryo sac is called hypostase. Then what is epistase? The thick walled cells found above the micropylar end, above the embryo sac is called epistase.